Hi, hello, welcome to my channel Mother Talks. Today my topic is nutritional requirement of bacteria and nutrient media. Objectives: to understand nutritional requirements of bacteria, or to study nutritional classification of bacteria, or to study constituents of nutrient media, or to understand types of nutrient media, to understand uses of different nutrient media. Nutritional requirement for cells is every organism must find in its environment of all of the substance required for energy generation and cellular biosynthesis. The chemicals and elements of these environment that are utilized for bacterial growth are referred to as nutrients or nutritional requirement. Many bacteria can be grown in the laboratories in culture media, which are designed to provide all the essential nutrients in solution for bacterial growth. The requirements for growth: physical requirements are temperature, pH, oxygen, hydrostatic pressure, and osmotic pressure. Chemical requirements for nutritional growth is carbon, nitrogen, sulfur, and phosphorus. Trace elements are oxygen, organic growth factor. The nutritional requirements of bacteria: first, nutritional requirement of all living organisms are similar. First one, source of energy: chemotrophs, organisms which obtain energy by oxidation of chemicals. Examples: Nitrosomonas europaea, oxidation of ammonium ions; E. coli, oxidation of glucose. Phototrophs, organisms which obtain energy from light. Example: Rhodospirillum rubrum, Chromatium ocani. Second one, source of elements. First one, lithotrophs. Lithotrophs are the bacteria which obtain electrons from reduced inorganic compounds as a source of energy. Chemolithotrophs are the bacteria which obtain electrons from chemical compounds. Nitrosomonas europaea. This is the chemical formula of chemical reaction of chemolithotrophs. Photolithotrophs are the bacteria which obtain electrons from ligand or gas. Chromatium ocani. Source of electrons or organotrophs are the bacteria which obtain electrons from reduced organic compounds. Chemoorganotrophs. E. coli. These are the chemical reaction of chemoorganotrophs. Photoorganotrophs or Rhodospirillum rubrum. Third one is source of carbon: structural organic molecules and energy source. Autotrophs are the organisms which utilize carbon dioxide as their major or even sole source of carbon. Nitrosomonas europaea, Chromatium ocani. Heterotrophs are the organisms which utilize organic compounds as their major or even sole source of carbon. E. coli. Rhodospirillum rubrum. Source of nitrogen is all organisms require nitrogen in some form for cell components in amino acids and proteins. Most bacteria decompose proteins, nitrogen gas or azidobacter and rhizobium. Inorganic nitrogen compounds are nitrates, nitrites, ammonium salts. Organic nitrogen compounds are amino acids and proteins. Oxygen, sulfur, and phosphorus are oxygen. Water molecular oxygen component, atoms of various nutrients needed for enzyme synthesis and energy production. Sulfur element: sulfur, inorganic sulfur or organic sulfur, needed for synthesis of cysteine, cysteine, methionine. Phosphorus, supplied phosphates, needed for synthesis of nucleic acid phospholipids. Metal ions: Ca++, magnesium plus potassium, and ferrous, required for normal growth. Trace elements are zinc, copper, manganese, nickel, copper, molybdenum. B3+, required for normal growth. occurs as contaminants of other components required in very small amounts. Metal ions act as cofactor in enzyme reactions. Most bacteria do not require Na plus. Halophiles require 12 to 15% of sodium chloride for maintenance of integrity of cell wall. Archaeobacteria. Water. Bacteria like nutrients by absorption. Nutrients must be present as aqueous solution for absorption by bacteria. It is polar solvent. It dissolves and disperses cellular components. It produces suitable environment for metabolic reactions. It is reactant in many metabolic reactions. It provides resistance to sudden and transient temperature changes. Vitamins act as coenzymes in enzyme reaction. Most of the bacteria can synthesize their vitamins. Some bacteria cannot synthesize their vitamins and should be supplied in nutrient medium. Vitamins: thiamine B2, Bacillus anthracis, riboflavin, Clostridium tetani, niacin, nicotinic acid, Brucella abortus, pyridoxine, lactobacillus, biotin, leuconstock, mesenteridos, pantothenic acid, morganella morgani, folic acid, leuconostic dextranicum, col- cobalamin B12, lactobacillus, vitamin K, bacteroidesis melanogenicus, buffer to neutralize acid and maintain proper pH. Peptones and amino acids or phosphate salts may act as buffer. Physical factors required for bacterial growth. First one is pH. Optimum pH. The pH at which the microorganisms grow best is example pH 7. Most bacteria grow between pH 6.5 and 7.5. Molds and yeast grow between pH 5 and 6 according to their tolerance for acidity, alkalinity. Bacteria are classified as acidophiles, acid loving, grow best at pH 0.5 to 5.4. Neutrophils grow best at pH 5.4 to 8 points. Alkali phyllis, base loving, grow best at pH 7 to 11.5. Temperature. According to their growth temperature ranges, bacteria can be classified as Psychrophilus 0.0 degrees Celsius and also grow best at 15 to 20 degrees Celsius. Psychotrophus grow best between 0 degrees Celsius and 20 to 30 degrees Celsius. Mesophils grow best at 25 to 40 degrees Celsius. Thermophils grow best at 50 to 60 degrees Celsius. Typical growth rates and temperatures. Minimum growth temperature, lowest temp at which species can grow. Optimum growth temperature at which the species grow best. Maximum growth temperature, highest temperature at which growth is possible. Food preservation temperatures. Temperature in this range destroy most air or microbes, although lower temperatures take more time. Very slow bacterial growth at 50 to 60 degrees Celsius. Rapid growth of bacteria, some may produce toxins at 20 to 40 degrees Celsius. Many bacteria survive, some may grow at 0 to 10 degrees Celsius. Refrigerated temperatures may allow slow growth at 0 degrees Celsius of spoilage bacteria, very few pathogens. No significant growth below freezing at minus 10 to 30 degrees Celsius. The danger zone in Fahrenheit is 60 to 120 degree Fahrenheit, in Celsius 20 to 50 degrees Celsius. Third one is oxygen. Aerobes required oxygen to grow. Obligate aerobes must have free oxygen for aerobic respiration. Example, Pseudomonas. Anaerobes do not require oxygen to grow. Obligate anaerobes killed by free oxygen bacteroids. Microaerophilus grow best in presence of small amount of free oxygen. Capnophilus carbon dioxide loving organisms that thrive under conditions of low oxygen. Facil- Facultative anaerobes carry on aerobic metabolism when oxygen is present but shift to anaerobic metabolism when oxygen is absent. Aerotolerant anaerobes can survive in the presence of oxygen but do not use it in their metabolism. Obligate. Organism must have specified environmental conditions. Facilitate. Organism is able to adjust to and tolerate environmental conditions but can also like in other conditions. Patterns of oxygen use. This is the picture of patterns of oxygen use. Fifth one is osmotic pressure. Environments that contain dissolved substances and exert osmotic pressure. Hyperosmotic environments, cells lose water and undergo plasmolysis, shrinking of cells. Hypoosmotic environment, cell gain water and swell and burst. Halophilus, salt loving organisms which required moderate to large quantities of salt, sodium chloride. Membrane transport system actively transports sodium ions out of cells and concentrate potassium ion inside. The Great Salt Lake in Utah. Nutrient media. Definition. The nutrient media is defined as balanced mixture of nutrients dissolved in water. Nutrients media are used for cultivation of microorganisms in laboratory. Many nutrient media are used to facilitate isolation, identification and enumeration of bacteria. Types of culture media based on their consistency: solid media, liquid medium, semi-solid medium. The second one based on the consistency and ingredients: simple medium, complex medium, synthetic or defined medium and special medium. The first one is special media. Under special media, enriched media, selective media, indicator media, differential media and transport media based on oxygen requirement, aerobic and anaerobic. First one is solid media. Contains 2% agar, colony morphology, pigmentation, hemolysis. Example, nutrient agar and bloody agar. Liquid media, no agar. This media is used to obtain bacterial growth from blood or water when large volumes have to be tested. For inoculum preparation, for preparing bulk cultures of antigen or vaccine. Blood culture for the isolation of pathogens from a mixture. Example, nutrient broth. Semi-solid medium or 0.5% agar or less. It has a soft custard-like consistency and is useful for cultivation of microaerophilic bacteria H. pylori or for determination of bacterial motility. Example, Wilson Blair media for Salmonella typhi. These are the pictures. Simple media or basal media. Example, NB consistency of peptone 5 grams, yeast extract, beef extract 3 grams and water 1000 ml. Nutrient broth plus 2% agar is equals to nutrient agar. Peptone 5 grams, yeast extract or beef extract 3 grams, agar 2 gram, water 1000 ml. Agar powder, it is a polymer of sulfuric acid ester of D-galactose. It is a polysaccharide obtained from seaweeds like Pteroclada geladium, brown algae. It is used as solidifying agent. 1.5 to 2% agar, solid medium. 0.5% agar or soft agar.
Used for preparing solid medium obtained from seaweeds. No nutritive value. Not affected by the growth of bacteria. Melts at 98 degrees Celsius and sets at 42 degrees Celsius. 2% agar is employed in solid medium. It can be used for cultivation of even thermophilus. It does not provide nutrients to bacteria and does not changes composition of media. It is not hydrolyzed by most of the bacteria. Peptone prepared from peptones like meat, cassia, gelatin, proteins or partially digested by acids or enzymes. Example pancreatic digest of casein, soybean digest. Use source of organic nitrogen and some amounts of carbon and nitrogen may contain vitamins and carbohydrates depending on kind of protein used can act as buffer. Meat extract and beef extract. It is aqueous extent of lean beef tissue concentrated to paste or powder. Use contains water soluble substances like carbohydrates, organic nitrogen, vitamin and minerals. Yeast extract. It is aqueous extract to of yeast cells available as powder. It is very rich source of B vitamins. It also contains organic nitrogen and carbon compounds. The second one is complex media, raw material or crude ingredients. Extract composition is not known, not reproducible, cheaper, easy to prepare, contain ingredients like peptone, meat extract, yeast extract and agar powder. Use cultivation of heterotrophs. Complex media other than basal media, they have added ingredients, provide special nutrition. Synthetic chemically defined media, also known as synthetic media. Pure chemicals. Extract chemicals composition is known, reproducible, costly. Use cultivation of autotrophs, metabolic studies of heterotrophs. Example of chemically defined media for cultivation of E. coli. Enriched media. It is rich in nutrients. Additional nutrient sources like blood, serum, plant or animal tissue extract or added to medium. Example blood agar, chocolate agar. Enriched media. It is a liquid medium containing selective agents. It all allows growth of specific organism and do not allow growth of unwanted organism. It is used to increase a relative proportion of desired bacteria in mixed culture. Example selenite F broth, tetraethanoate broth for salmonella. Tetrathionate inhibits coliforms while promoting typhoid and parathyroid bacilli. Selective media. It is a solid media containing selective or inhibitory agent. If inhibitory agent is added to enriched media, then the media becomes selective. It allows growth of desired microbacterium and does not allow or inhibit growth of unwanted microorganisms. Example McConkey's agar, intestinal bacteria, mannitol salt agar, Salmonella aureus. Differential medium. This medium helps to differentiate between two groups of bacteria growing on it. McConkey's agar differentiates between lactose fermenting and lactose non-fermenting bacteria between bacteria. Lactose and neutral red together form indicator system. Example McConkey's medium consists of peptone, lactose, agar, neutral red and taurocolate, which shows lactose fermenters in pink colonies while non-lactose fermenters in colorless or pale. Lactose fermenters are E. coli, non-lactose fermenters are Shigella and Salmonella. Nalgar's medium enables us to view lethinase activity. Colors of colonies of lactose fermenters and non-lactose fermenters on McConkey's agar. A. lactose fermenting bacteria and B lactose non fermenting bacteria. Blood agar differentiates between alpha and beta hemolytic bacteria. Alpha hemolysis refers to p- partial lysis of RBC showing green color colonies. Example Streptococcus viridans. Beta hemolysis refer to complete lysis of RBC showing colorless. Example Streptococcus pyrogens. Indicator media. These medium consists small chemical or indicator which changes color bacteria grows in them. Example Wilson and Blairy medium. It contains ferrous sulfate, salmonella reduce sulfate to sulfide and forms black sheen on colony. Cyanobacterium diphtheria produces black colonies or McLeod medium due to reduction of potassium tellurate or tellurate to metallic tellurium. Media for enumeration of bacteria. It is used to determine total viable count of bacteria present in natural soil, water, foods or sample. It is most nutritive than simple nutrient agar and supports growth of most of the bacteria present in natural sample. Example standard plate count medium. Maintenance media. These are required to maintain viability and psychological characters of culture. They are not supposed to support rapid growth of bacteria. Rapid growth may result into rapid death of bacteria. Chemicals like glucose should be avoided in this media. Transport media. They are used for temporary storage of patient sample being transported for cultivation to laboratory. It is used to prevent death of microorganisms during transport and to maintain viability of all organisms in specimen for sufficiently long period. Contains mainly buffer and salts. Stewart medium soft agar gel containing reducing agents to prevent oxidation and charcoal to neutralize Neisseria gonorrhea. Buffered glycerin medium E. coli. Anaerobic medium. This medium is used for cultivation of anaerobic bacteria. It contains reducing agents. Example Robertson's cooked meat medium. Fluid thioglucolate medium. Thioglucolate medium. The medium supports the growth of all the organisms with varied oxygen requirements. The medium is boiled before used in order to eliminate oxygen. Obligate aerobes grow only at top of the medium. Microaerophilus in the middle and anaerobes will grow only at the bottom of the medium. Composition is yeast extract, acetone, sodium chloride, L-cysteine, thioglycolic acid, agar, methylene blue and deionized water. This is the picture of methylene blue indicator strip, anaerobic jar and gas pack. Bacterial growth curve. During typical bacteria growth, bacteria cell divide by binary fusion and their mass and number increase in an exponential manner. Bacteria growth in culture can be separated into at least 4 distinct phases. In between each phase there is a transition period. T represents the time required for all cells to enter the new phase of the growth curve. When log number of cells is plotted against the time growth curve is obtained consisting of 4 phases: lag phase, log phase and exponential phase, stationary phase and phase of decline. In this graph we can see that lag phase, log phase, stationary phase and death phase. In this graph, the lag phase is at starting of the graph. The log phase is where the graph is increasing. The stationary phase is that the the middle phase of the graph, and the death phase is the end of the graph. This is another picture of logarithm. The lag phase. The addition of inoculum to a nutrient medium is not followed by doubling of the population. They are metabolically active, grow in size, synthesize enzymes and incorporative molecules from medium, produce large quantities of energy in the form of ATP. Factors affecting the length of lag phase is that conditions of microorganisms and nature of media. The log phase is that organisms have adapted to a growth medium. Growth occurs at an exponential log rate. The organism divided at their most rapid rate at a regular genetically determined interval. The time required for the population to double is called generation time or doubling time, which is given by the equation where g is equals to generation time, number of generations n at a particular time t. The log phase during exponential growth, the growth rate termed r is the reciprocal of generation time, number of generations n at a particular time t. In this phase, bacteria are overfed with the nutrients. The number of cells generate than the number of cells die. Stationary phase is that several hours after the inoculation is called as stationary phase. In this stationary phase, there are the four things that happen in the stationary phase is that the cell division decreases to a point that new cells are produced at same rate as old cell die. The number of new cells produced is equals to the number of old cells died. The number of live cells stay constant. The population remains constant for a time. Supply of nutrients is decreased. Decline or death phase. Following the stationary phase, the medium becomes less and less supportive of cell division. The cell lost their ability to divide and due to depletion of essential nutrients and accumulation of inhibitory products such as acids, number of viable cells decreased at a logarithmic rate. The death rate contain continuation of the death rate is reproductive rate. Synchronizes growth, a hypothetical situation in which the number of cells in a culture would increases in a stair step pattern, dividing together at the same rate. If all the cells in a culture medium are in the same stage of the growth cycle, then they are said to e-growing synchronously. A population can be synchronized by manipulating the physical and chemical environments for X. The cells may inoculated into a medium at a suboptimal temperature such that they will metabolize slowly but not divide. When the temperature is subsequently raised, all the cells undergo synchronized division. Next, the non-synchronized growth is a natural situation in which an actual culture has dividing at one rate and other cells dividing at a slightly slower rate. This is the graph of synchronizes and non-synchronizing growth. Preparation of media. The preparation of bacteriological media usually involves the following step. The preparation of media is having the 5 steps. The first one is each ingredient or the complete dehydrated medium is developed in the appropriate volume of distilled water. The second point in the preparation of media is that the pH of the fluid media is determined with a pH meter and adjusted if necessary. The third point of preparation media is that if a solid medium is desired, agar is added and the medium is boiled to dissolve the agar. The fourth point of the preparation of media is that the medium is dispensed into tubes or flask. The fifth point of preparation of media is that the medium is sterilized generally by autoclaving some media or specific ingredients that are heat labeled or sterilized by filtration. Cultivation of aerobic bacteria. To grow aerobic or facultative bacteria in tubes or small flasks. Incubation of the medium under normal atmospheric conditions is generally satisfactory. However, when aerobic organisms are to be grown in large quantities, it is advantageous to increase the exposure of the medium to the atmosphere. This can be accomplished by dispensing the medium in shallow layers for which special containers are available. Aeration can also be increased by constantly shaking the inoculated liquid cultures. Cultivation of anaerobic bacteria. Stringent anaerobes can be grown only by taking special precautions to include all atmospheric oxygen from the bacteria. Such an environment can be established by using one of the following methods: pre-reduced media. During preparation, the culture medium is boiled for several minutes to drive off most of the dissolved oxygen. A reduced agent, example cysteine, is added to further lower the oxygen content. Oxygen-free N2 is bubbled through the medium to keep it anaerobic. The medium is then disappeared into cubes, which are being flushed with oxygen-free N2 stoppered tightly and sterilized by autoclaving. Such tubes can be stored for many months before being used. During inoculation, the tubes are continuously flushed with oxygen-free CO2 by means or a cannula stoppered and incubated. This is the anaerobic jar inoculation loop. Gram positive and gram negative bacterial chat. And thank you. Please subscribe my channel for more updates and more interesting videos.